Assalamualaikum to those who have just joined, and we are going to begin from microtubules. We already covered it in the previous week, but due to some technical glitch, we were unable to cover it in the live class. So, starting from the concept of tubulin, tubulin is a protein, okay, which makes up microtubules. So, what exactly microtubule is? First we need to cover up the composition of how microtubules are made, and then we'll proceed towards the function of microtubules. Okay. So, okay, there's a protein named as tubulin. Okay. And there are two categories of tubulin. One of them is alpha tubulin, and other one is beta tubulin. So I'll use two different markers to differentiate between the concept of alpha and beta. Okay. So this is an alpha tubulin. Okay. Alpha tubulin हो गया ये, and this is your beta tubulin. So tubulin क्या है? Tubulin is a protein, and alpha tubulin, beta tubulin, their what do we say? Their sub circles make up dimer. Okay. So when beta tubulin combines with alpha tubulin, it makes a dimer. Okay. So this is the beta one, this is the alpha one, and together they'll make a dimer. So far are we clear? अभी तक concept clear हुआ? मैंने क्या बोला? I said that tubulin has two categories, alpha tubulin and beta tubulin. And when alpha tubulin and beta tubulin combine, they form a dimer. Okay. So then dimer forms protofilament. Dimer, when we combine lots of dimers, then they form protofilament. Okay. So consider dimer as the monomer and consider protofilament as the polymer. Do not write this in your exam. I'm just trying to explain the concept of dimer and protofilament. So dimer is made up of alpha, beta, then many dimers, they join up and they form protofilament. Okay. So they form protofilament. And when अच्छा ठीक है, प्रोटोफिलामेंट इज अ सिलिंड्रिकल लॉन्ग ट्यूब्यूल या सिलिंड्रिकल लॉन्ग ट्यूब. ठीक है. सो डायमर जब बहुत सारे कंबाइन हुए तो उन्होंने प्रोटोफिलामेंट बना दिया. And then when protofilament will combine, so 13 such protofilament need to combine, and when they combine in the cylindrical manner, they form a microtubule. So dimer, alpha beta will form a dimer. Dimer will form a cylindrical protofilament. It's a tube-like structure, and when only 13 protofilament, not many, when 13 such cylindrical tubes will combine, they'll form microtubule. Okay. So far, are we clear on the structure of microtubule? I haven't discussed the function of microtubule so far, so I hope we are clear on the structure of how microtubules are made. Clear?
Yes, miss.
All right. So let me show you the picture of microtubule, and then we'll proceed. So microtubule basically, see, this is not a very clear picture, but we can understand that this is a cylindrical structure, and 13, if we'll count, then these cylinders will be 13 in number, and this is a hollow structure, right? So the diameter of microtubule, you need to remember the diameter of microtubule. This is an important component of your paper. This is 25 nanometers. So the diameter of microtubule is 25 nanometers, and protofilaments are arranged in such a way that it will make a hollow structure of microtubule. Okay? Achha ji. So it's also written here that it is 25 nanometers in diameter. Then, so now what exactly is the function of microtubules? So microtubules are responsible to make cytoskeleton of the cell. See, we also have a skeleton. We have bones that have made our bodies, right? So cell also needs some structural assistance in order to maintain its shape. So that structural assistance is given by cytoskeleton, all right? So cytoskeleton, and what exactly cytoskeleton is made up of? It is made up of microtubules. So cyto means cytoplasm, and skeleton means the skeleton that we also have. So cytoskeleton is the skeleton of cytoplasm, which provides support and movement to the cell. So far, are we clear?
Yes, miss.
Okay, then we have centrioles. Centrioles is another important organelle, and it is only found in animal cells. It's not found in plant cells. So keep this as a difference between plant and animal cells. That it is only found in animal cells. Then again, microtubules are responsible to make centrioles as well, along with the cytoskeleton. So centriole is also made up of microtubules, and as far as this function of centrioles is concerned, we'll discuss in the chapter of mitosis. Okay? So, Abhi, if you're unable to understand spindle fibers, then it's completely fine because I'm not going to explain it any further. You just need to remember that it is organizing spindle fibers during cell division. So, this is a very detailed topic, and we cannot just go through a simple line. So, as far as this topic is concerned, you just need to remember this line and you're good to go because you'll cover this in a great detail in the chapter of mitosis. Okay, so we are skipping it for right now. We are skipping it right now. We'll cover it in mitosis. Achadi, next we have cilia. Cilia and microvilli. Okay, so a quick check. Do you remember the difference between cilia and flagella? What was flagella responsible for?
For the movement.
For the movement of the prokaryotic cell. All right. So the difference between cilia and flagella, one major difference is that cilia are also found in animal cells. So we also have cilia mainly in our respiratory tract, and we need to remember how they look like. So they look like here, like projections, and they're also made up of microtubules. So you need to make a chart that microtubules are responsible in the formation of cilia, they're responsible in the formation of cytoskeleton and in centriole. So far, we have learned three organelles that are made up of microtubules. So you can make a small note in your notebook in order for you to not forget it by the end. All right, so cilia here like projections, they are also made up of microtubules, and cilia also has a similar function like flagella. It also allows the movement. So it is also a thread-like structure. So it helps in sweeping up the substances over the cell, and they're mainly present in respiratory tract to sweep up the mucus. So they are the hair-like projections, thread-like projections that will help to sweep up the mucus or any particle which is not needed in the body. Okay, and then we have microvilli. Microvilli is okay. If this is a cell, I don't know what type of cell I made, but let's consider that it has microvilli. These are small finger-like projections. So these small finger-like projections, they are present over the cell membrane, and they're mainly present in our GIT. They're mainly present in small intestine, so they increase the surface area for absorption of different food materials. Okay. Then we have cell wall. We know that cell wall. Okay, quick check. Cell wall in plant cell is made up of.
Cellulose.
Excellent. And what about bacteria? Cell wall in bacteria is made up of.
Peptidoglycan,
And what about fungi?
chitin,
Excellent. Okay. So cell wall provides structural support to different types of cell, and you need to remember the composition. Okay, we already discussed it. Next. So now moving towards the organelles of plant cell mainly. Plant cell has chloroplast, one of the vital organelles present in plant cells because chloroplast is responsible for photosynthesis, and photosynthesis is the process of making food for plants. So you need to remember a few specifications. Okay, so there is one question that comes repeatedly in your past papers. It is between the differences and the similarities between chloroplast and mitochondria. Okay, so I'll highlight the differences and the similarities of chloroplast and mitochondria. If you want, you can note it down. Okay, so one of the similarities between chloroplast and mitochondria is that it has double membrane. We already talked about the organelles that have double membrane. They were chloroplast, they were nucleus, and mitochondria. Okay, so it also has double membrane with circular DNA in it. So this is also one of the similarities between chloroplast and mitochondria, that they have circular DNA. Then the difference between chloroplast and mitochondria is that they are membrane-bound compartments and they contain thylakoids. Okay, so now I'm going to explain you the difference between thylakoids and grana, because let me see if we have a picture of chloroplast over here. I believe I haven't put up the picture of chloroplast. because we do have a chapter of plants. I think we have a chapter of plants. It is, I think, chapter number 7 or so. So we'll discuss it there as well. But in order to explain you, I can tell you one thing that okay. So chloroplast has thylakoids and grana. Okay, if this is a chloroplast, let's visualize it. So this is a chloroplast with double membranes. Okay, and it has a circular DNA. So this is the internal structure of chloroplast. Okay, so you'll see number of projections like this within the chloroplast. I'm sorry for my drawing because I'm not very good at drawing. But I think we will be able to deliver you the concept of thylakoid and grana. Okay. So, actually, have you ever seen a pile of coins? Yes or no? Pile of coins. Um, Okay, obviously you have seen the pile of coins. अच्छा जी, अच्छा. Let's consider that. Um, how do I make you explain this? अच्छा, let's consider that you have a pile of coins. ठीक है? This is a pile of coins. Do not consider this as thylakoid that I've just made. Consider this as pile of coins. So this pile of coins is known as grana. Okay? This pile of coin is known as grana. Okay? It will be better if you write it along with me in your notebooks. Pile of coins is known as grana. Okay? And one single coin, if I take one coin out of this stack, this single coin will be known as thylakoid. Okay? Thylakoid. The single coin will be known as thylakoid, but a pile of coins will be known as grana. You want समझ आई? Okay. So I'm trying to say that thylakoid and grana, they are the same things, okay? But a collective stack of thylakoid is known as grana, while a single coin is known as thylakoid. And what are they responsible for? You do not have to cover this right now. You'll be covering in-depth function of thylakoid, grana, chloroplast in your A2s. So let's not go there because then we'll have a lot to discuss. But if you're clear on this, if you're clear on the difference between thylakoid and grana, then you will be able to label it in your then you'll be able to label it in your papers. Because you are supposed to label these components of chloroplast in your paper. Okay, so agar they mark something like this, then what you'll call it? What will you label this?
Grana.
Grana, excellent. And if they label just if they label just one coin, then you'll call it thylakoid. Okay. So you need to remember the difference between grana and thylakoid. Then moving towards plasmodesmata. अच्छा ठीक है. So plasmodesmata is mainly found in plant cells, and I'm making 2 plant cells over here. Okay. Mm. Mm. Okay. So these are two neighboring plant cells, all right? And there's a passage between both of them. So this passage between both of the neighboring cells is known as plasmodesmata. And why do we need plasmodesmata? It is because these are narrow junctions. It's a very minute junction basically, and it allows the movement of substances from one cell to another cell. Okay, so this is just a minute passage between two plant cells. Never write that you can find plasmodesmata in animal cells. This is just a property of plant cells. All right, then we have vacuoles. We already talked about the major difference between vacuoles in animal cell and in plant cell. That we have one solid permanent vacuole in plant cells, and the membrane of vacuole in plant cells is known as tonoplast. And while the animal cells, they have very small vacuoles, they contain cell sap, CellSap is basically it is the nutrient of a cell. It contains proper nutrients. It also contains waste materials. So it acts as a reserve of nutrients for plant and for animal cells. Now, moving towards ATP. So ATP is adenosine triphosphate, and we know that it is the energy currency of the cell. So it is also produced by mitochondria. And so basically, let's understand the concept of currency. Okay. So in Pakistan, we only accept PKR, right? But we cannot give a PKR rupees to a shopkeeper in America, right? So the currency in Pakistan is PKR, while the currency in USA is dollars. So in order for cell to utilize energy, it needs a molecule in the form of ATP. It does not need food that we are consuming, right? So our food has to convert in something like a molecule of ATP. Only then the cell will be able to and only then the cell will be able to consume energy. Okay? So cell is not going to understand what glucose is, what proteins are, what lipids are. The cell will only understand the molecule of ATP. Okay. Either way, they are all the currencies of they are all the energy currencies, but the acceptable currency for cell is ATP. Okay. And ATP is used in processes like active transport. I'm sure you are familiar with the transport mechanism of active transport, but you have no idea about bulk transport. So the next chapter that we'll be starting tomorrow of cell membrane, we'll talk about bulk transport in that chapter. Okay. Then small part of viruses is also part of your chapter 1, that viruses are very simple in structure and they have a protein. Acha, let's draw virus. Okay. Because it's very simple to understand. So this is a protein code of virus, okay, which has enclosed the entire component of virus, and this is known as capsid. Okay, capsid is a protein in nature, and capsid is encapsulating the entire virus. Okay, do not write that this is the cell membrane or cell wall of animal cell or of plant cell. So only capsid is in circulating viruses, and inside it has a protein. Okay, and this protein is known as capsomere. Okay, so virus has a protein called capsid, and it has a protein called capsomere. Then a virus can have a DNA or RNA. So there is a choice between DNA or RNA in the viruses. So we can never write that viruses contain DNA and side by side they contain RNA, because different viruses have different composition of DNAs and RNAs. Then they are parasitic in nature, which means that when they're in the environment, they are not harmful. But once they gain entry in a living body, then they become parasitic in nature. And parasitic means that they are going to destroy the machinery of others, and they'll build their own machinery to produce viral particles. So basically, this happens that, for example, if you get influenza or something like that, then that virus is going to destroy your machinery, your protein making machinery, and it is going to make its own protein making machinery. And this is how viruses are going to survive. Okay? So we are done with this chapter, but an important component of this chapter is calculations. And I know that people who opt for biology, they are quite weak at maths. So this is not a journalized statement or something. I don't mean to offend anybody, but we need to cover this subtopic of calculations thoroughly, because you'll see this extensively in your paper 1s and also sometimes in paper 2. So we'll practice calculations a bit, and then we'll proceed towards the past papers.